Hey, Jay. Who? Who did? Oh.
Hey, Mrs. Marquardt.
Okay, well,
I had to go to the doctor's today
Mhm.
to get my nose cauterized.
Ah,
So I have a cotton ball up my nose.
okay. Well, if you start bleeding and you need to leave, you don't need to get permission, okay?
Got it.
Let's see. Okay, I shifted the plans a little bit due to the 2-hour delay today. We really need a full period for those of you that are working on the lab in person to do their part. So I'm going to share the agenda that I've actually finally created for this week. I'll be posting it after school today, but it'll give you a better guideline of what we're working through. So today, instead of starting the plankton lab, which I did post in Google Classroom, but we're not going to start it today, we are just going to finish the lesson 31 slides. So we're just going to take some more notes today, unfortunately. But then for the next basically 4 class periods, we'll be working through the lab. I still plan on giving the opportunity for A students and B students to do the lab portion in person. So it's going to run over, like I said, it's three class periods. It's going to run over 3 class periods. This is how it's going to look. Tomorrow, since we're all virtual and nobody can do the actual lab, there is a portion of the lab that is just all paper stuff, some background stuff. So everybody will work on part 1 tomorrow. And again, that's already in Google Classroom. I'll pull it up just so I can show you what that looks like. Then this is where it's going to look a little different depending on the group you're in. On Thursday, assuming we don't have snow, some of the students will be working on part 2, and those are the students that are in the building. So if you're a B student or a 4-day student, you will be doing the physical part of the lab. If you are all virtual or an A student, you will be finishing up any part of part 1 that you didn't get done yet. Okay, so you don't have to get all of part 1 done tomorrow. Friday then is a half day. So as of right now, I'm planning on just doing a very short mini lesson on harmful algal blooms. And then Monday's a holiday. So that would bring us to Tuesday of next week when we have our A students back in. And then those A students will do part 2 of the lab, and the virtual and B students will be finishing up their version. So what does this look like in Google Classroom? In Google Classroom, there's actually 2 different documents. One of the documents is for students that are all virtual. So if you will never physically be in the building to do the microscope portion, you have your own version. Part 1 looks exactly like the other version. There's a lot of pictures in here that aren't loading currently. Okay, looks like a lot of reading, but it's not. Few questions to answer. Okay, I didn't give you space to answer them here, but you can certainly just type and fill in as much as you need. Don't use this as a judge for the space for answering the questions. And again, you're basically going to have 1.5 full class periods to work on that. Then for those of you that aren't coming in person, you're just going to watch a video to refresh you on the microscope, figure out your parts in your microscope, which should be a review, and then a couple of simple questions from the video. Additionally, it's going to ask you to look at these images that would resemble some that we might see under the microscopes, and you're going to have to make some drawings. Okay, so your microscope section is really just going to be pictures. The way that differs then from the in-person is essentially that you're just going to be looking through the microscope to get your pictures. Okay, so part 1's the same. Part 2, you do still need to watch a short video just to refresh you on the microscope, but you don't have to answer any questions related to it. And then since it's a little harder to see the images in the microscope and a little more of a challenge for you, you won't have those microscope questions and you'll just have to do your drawings. Okay, so a little less writing work, a little more physical work for you guys. Okay.
Can I use my own microscope?
Are you in the building?
No.
What are you going to look at?
Okay.
Your toenail? Yeah, you could if you had a slide, but you don't. So unfortunately, that won't work for you. Okay. I have information here about timing, but again, just go by the agenda and we'll talk through it each day. So, okay. So for today, we're not doing anything with that. Tomorrow, everyone is doing part 1, but we'll join by Zoom and just remind you of that tomorrow. All right, so let's, whoops, I didn't mean to do that. Let's finish up our notes on phytoplankton. That way you'll have a good background when you do the slides. Why it's being slow. Come on. Huh. There we go. All right, so we had just finished discussing the diatoms, which were one of the 6 kind of main groups of phytoplankton. Okay, so this was just a picture we showed of how diatoms can chain together to form colonies. This is a bunch of different organisms instead of one big organism. and that they can use some kind of mini jet propulsion system to kind of swim. So diatoms are very interesting because they can actually reproduce sexually and asexually. Remember that asexual just simply takes the organism and splits it in two. The DNA doesn't change at all. You don't have any crossover happening or any mixing of DNA. So it is a very effective process for reproducing quickly, and it's why we can get very large numbers of diatoms in the ocean. The asexual form of cell division is called vegetative cell division. The only problem with this asexual cell division in diatoms is, if you recall, we said that their two frustules were like the two halves of a petri dish. And if you remember from being in person, in a petri dish, one of the halves is a little bit bigger than the other so that it can sit over top of it. Okay, so when they asexually reproduce, they split into two halves, and then the new shell will build inside the old one. Okay, so as it splits again and again and again, the shell gets smaller and smaller and smaller because the new shell is always getting built inside the old shell. So eventually, this vegetative reproduction leads to diatoms that are extremely small and not very effective anymore. So although it produces a lot of organisms, they're not all large organisms. Okay, but there is constant asexual production happening. Additionally, though, and this is one of the reasons why diatoms have been able to survive so long, is because they can also sexually reproduce. There is a sperm and an egg. They do have crossover of genetic material so that there's changes in the daughter cells, and you're not having the constant DNA. So when large changes in the environment happen that may have wiped out entire species, the diatoms were able to adapt by mutation and other things. So they can not only reproduce quickly, but they can also survive harsh conditions because of the ability to sexually reproduce. What does their sexual reproduction look like? Well, essentially you have a sperm cell, it merges with the egg cell, and then you have a new cell which then can divide from there. So not extremely fancy, but it works. So just to show you how quickly a cell can reproduce through asexual reproduction, it's a logarithmic process. In other words, it happens faster and faster and faster. So on day 1 or 0, you might have one cell, then it goes to 2, then it doubles, 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 doubles. So when it keeps doubling, we call that logarithmic growth. And while the process starts out gradually, it can be explosive, okay? And that's also why you can get extreme diatom blooms, because they can reproduce so quickly. Okay, we're moving now to our last phytosynthetic organism, and this is called the dino or dinoflagellates. Now, a lot of people want to say this is an animal or a zooplankton. They see that word dino or dino and they're like, oh, dinosaur, or, you know, it's an animal. But no, these are still phytoplankton, okay? They're a little different than some of the other phytoplankton. First of all, they're very complex. They're the most complex single-celled organism out there. In fact, they have 70 times more sets of chromosomes than human beings. So they have a very complex genome, which allows for a lot of genetic variation. They are protected by chitin-like plates. So it's not really a shell, it's more almost like a leathery material. So that protects them. They call that an armor. But it is possible to have what they call naked dinoflagellates, which don't have any type of shell or armored plates. All dinoflagellates have two flagella. Okay, remember those are the appendages that they can use to swim. One of them wraps around the body, and it's called the transverse flagellum. So what this allows to happen is when they squiggle this transverse one that wraps around their body, it causes them to spin. So they will move in a spinning motion. And then their rear flagellum, called the longitudinal flagellum, will be what propels them forward. So they have a very interesting, quote unquote, swimming pattern. So while all algae have some type of propulsion, well, not all of them, but while most phytoplankton can propel themselves in some way, these would be the closest to looking like swimmers that are out there. One of the most interesting things about dinoflagellates is only about 50% of them actually have chloroplasts, which means that only 50% of them can do primary production on their own. So you wonder then, well, why are they considered phytoplankton if half of them can't do photosynthesis? Well, it's because of the remaining half do something called symbiosis. I'm going to pause this video for a second. And symbiosis is when they utilize another organism to basically help them do something they can't do. So the non-photosynthetic dinoflagellates will actually engulf or ingest a cyanobacteria that can photosynthesize. And once they ingest that, it essentially acts like their photosynthetic organelles. And they believe that this is actually how eukaryotic organisms first formed, was by ingesting these prokaryotic organisms and making them part of their body structure. So here today, we still see this happening. And by doing that, then they are able to create their own food through the ingestion of these cyanobacteria, these prokaryotic blue-green algae. All right, so this just shows what some of these little guys look like moving around in the water. And this particular example shows all of this particular type that has these cyanobacteria in them. But you can see how they look like they're swimming. Okay, the other interesting thing about dinoflagellates is most of them can bioluminesce, meaning they can undergo a chemical process in their bodies that allows them to give off light, just like lightning bugs. Okay, so lightning bugs also glow because of bioluminescence. This is actually visible here up north if you go to the coast, but because most of the northern coastlines are so well lit, it's not easy to see. Particularly, you can see that in the summertime. But if you go into some tropical locations, it's pretty fascinating because even when the waves come in, you can see the phosphor or the bioluminescence happening. Okay, one last thing, and this is actually pretty important because it talks about why phytoplankton grow where they do, and a lot of this should be a review, so hopefully it's just re-cementing what we've already talked about. So light definitely limits where phytoplankton can live because they go through photosynthesis. They need to live where there's light available. So they cannot live below that euphotic zone. They must live near the surface. So how do they do this? Because anything as small and as light as it is, if it doesn't have adaptations, is going to eventually sink. So some of the adaptations that phytoplankton have evolved to keep them from sinking quickly in the water column are the following. Number one, their shape. So they're small, which certainly keeps them from sinking fast, but also by forming like webs and colonies, they'll have more surface area so they won't sink. And also they'll build like silica spines and things to give them more of an area to float. So just like when you float on your back, you float better than if you tuck into a ball. The same thing happens with the phytoplankton. Okay, we said about the smaller cells, they definitely sink slower. We talked about how diatoms can actually produce oils to help them stay buoyant. And of course, we know that some of them have a weak swimming behavior because of those flagella and or jet propulsion. So that's some way they overcome the problem of sinking so that they can stay in the light. The other thing that we know is important to life is having nutrients. And we know that most of the nutrients are at the bottom, whereas the phytoplankton must live at the surface. So for this reason, you're going to have your greatest phytoplankton growth in areas where the nutrients from the bottom get pulled up to the top. So things like in winter turnover, that'll happen, and then in areas of upwelling, either Ekman upwelling or equatorial upwelling. So just to show you how nutrients can limit the growth, this graph shows that the blue line has the most nutrient concentration, and you can see how much growth there is in the phytoplankton versus the red line, which has the least amount of nutrient in the water. Okay, so obviously nutrients affect growth. Okay, next slide we don't need. It talks about cell size and surface volume to area ratio, but you had that in bio. So, okay, how do we collect phytoplankton samples? Well, with a net. So the best way to collect your phytoplankton is simply to drag a very fine mesh net beside or behind the boat as you travel. It allows the water to pass through, but it will collect the phytoplankton. And there's actually a little container at the back that will collect the actual solid material, which is your phytoplankton. So they can collect large numbers of phytoplankton to study this way. But you can also collect them the old-fashioned way, simply dropping in a water bottle and bringing it to the surface to collect. But that would be a less effective way to do it. All right, last slide. So this shows a measure of the chlorophyll. Okay, so showing how much photosynthesis there is in certain areas. And I want to point out, again, why their concentrations are greater in some areas than others. So the red represents high concentration of chlorophyll, the blue would be low. So a lot of production in your red areas, not so much in your blue. Remember that along certain coastlines, based on the way the winds typically blow in that direction, you can have Ekman upwelling. So where there are areas of Ekman upwelling, you're going to have a lot of growth of phytoplankton. In these blue areas, which are showing not a lot of growth, these are your tropical, or these are your gyre areas. So like the Atlantic gyre, and again, no upwelling occurs in these areas, so you're not going to see phytoplankton. And then of course in your upper water bodies, this is where the entire water column is the same temperature. So it allows for that convective mixing at all depths. And that's why we also see production of phytoplankton at those higher latitudes. Okay, so it all ties in, it all comes together. And as we move through the organisms, you're going to see this more and more. Okay, that is it for today. So tomorrow then, we'll meet quickly. I'll remind you what your Responsibilities are, if you're bored tonight and want to get started on part 1, you're welcome to, but it's certainly not a requirement. Okay? I'll see everyone tomorrow.